Okay, so we are going to summarize what happens in lysogenic versus lytic cycle in bacteriophages. Just briefly describe what bacteriophage is. So the virus that infects the bacteria are referred to as bacteriophage or phage for short. There are different kinds of phages, lysogenic versus lytic phages, depending on what kind of life cycle they go through. So the first step is always the same. The phage or the bacteriophage attaches to the surface of the bacteria. And when it attaches, it will inject its DNA chromosome into the cytoplasm of the host. In this case, our host is bacteria. Remember, bacteriophages are infecting the bacteria. When the DNA gets into the cytoplasm, it becomes it's circularized. At this point, the bacteria needs to decide whether to go through the lytic cycle or the lysogenic cycle. And environmental conditions and the health of the bacteria will decide on which way to go. If it is going through the lytic cycle, then this phage DNA will be replicated many, many times. And from that DNA, all the viral particles or viral components will be produced, and they will be all put together, assembled, making new viral particles. In one bacteria, there would be around 200 newly made bacteriophages ready to be released into the environment. So, there are so many bacteriophages assembled in, and it's so much the bacteria now lyses, releasing all the viral particles into the environment. So when we look at the lytic cycle, we are seeing at the end of the lytic cycle, the host cell is being destroyed. It's lysed. So any cell that lyses, that means it's dead. Okay? So at the end of the lytic cycle, the host cell dies, and there is a production of many bacteriophages that are being released into the environment, and each one of those viral particles are free to go and infect uninfected bacteria. This is the lytic cycle. So at this step, if the bacteria decides to go to lysogenic cycle, then the bacterial chromosome and the viral chromosomes fuse together through a recombination event, and the viral DNA becomes part of the bacterial DNA. And in this case, we refer to viral DNA as prophage. It's not a separate DNA anymore. It's part of the bacterial chromosome. That's why it's referred to as prophage. So, since it's part of the bacterial chromosome, every time bacteria divides, it needs to replicate its DNA. Every time it replicates its DNA, it's also replicating the viral DNA, because viral DNA is part of the bacterial DNA. It's not separate anymore. Once the bacterial DNA replication starts, it will just continue until everything is replicated. So this is how we are replicating or multiplying the viral DNA. It's being replicated, but no new virus is being made or released into the environment. Occasionally, that prophage will be excised from the bacterial chromosome and going back to the lytic cycle. But when we look at the lysogenic cycle, we are seeing that the host cell is not being damaged. It's not destroyed. That's one thing that's separate from the lytic cycle. And two is no viral particles are being made. Okay, so those are completely opposite of each other, lytic versus lysogenic cycle. In lysogenic cycle, we have two terms. Prophage is referring to the viral DNA that's integrated into the bacterial chromosome or the bacterial host chromosome. And another term is the lysogenic bacterium. This is a bacterium that contains the viral DNA in its chromosome. Okay, so the phage, if the phage is lytic, it will only go through the lytic cycle. But if the phage is lysogenic phage, it can go through the lytic versus lysogenic cycle. So depending on its health, it can go through the lytic or lysogenic cycle. In the lysogeny, you are spreading the virus without killing the host cell. So the viral DNA is there, but viral particle is not being made. Phage genes that are embedded in the bacterial chromosome can cause the production of toxins or the enzyme that causes the pathology of the virus. So this is called the lysogenic conversion. We have a couple of really pathogenic bacteria, and they have the viral genome embedded in their chromosome. Those are lysogenized bacteria: Corynebacterium diphtheria, Vibrio cholerae, Clostridium botulinum, and Streptococcus pyogenes. So all of those have toxins that are coded by the viral genome that is embedded in their chromosomes. Because those viral DNA provides toxicity or increases the virulence of the bacteria, it's beneficial for the bacteria, and the bacteria will keep them in their chromosome.